Let's talk about immunization. There are two different categories: active immunization and passive immunization. Active immunization is the result of stimulating a person's immune system produce antibodies. As a result, memory cells are formed, and this confers immunity over a relatively long time, in many cases, for the rest of your life. There are two categories of active immunization: natural and artificial. Natural active immunity is achieved by recovering from a particular disease. In this case, you have to get sick, experience the symptoms, hopefully not come down with any long-term detrimental symptoms or death, and then recover from the disease. And finally, after all of that, you would have active immunity for the rest of your life in many cases. On the other hand, artificial active immunity is the use of vaccines. It's artificial in that it's intentionally administered into the body to provoke an immune response. These vaccines are designed to not cause disease, but still stimulate the immune system. So they are designed to have all of the advantages of active immunity without the disadvantage of being sick. Active immunity requires three basic components. The vaccine must be safe, effective, and stable. Vaccines can be made in one of three different ways today. The first way vaccines can be made is from what's referred to as live attenuated microbes. Live means that they're able to reproduce inside the body, and attenuated means that they're weakened. If they were just living microbes, then that would be like getting sick. But again, the idea behind vaccines is that they are safe and don't cause disease. So in order to be a vaccine, it must be attenuated or weakened. Live attenuated vaccines tend to produce the longest lasting immunity of all vaccine designs, because they can replicate in the body and engage the immune system much the same way as a natural pathogen would. However, because they're attenuated, they produce mild or no symptoms at all. With any live attenuated vaccine, individuals with impaired immune systems may have difficulty fighting the vaccine strain even though it's weakened. Examples of vaccines using the live attenuated microbes are the Sabin or oral polio, measles, mumps, rubella vaccine, rotavirus, and yellow fever vaccine. A second type of vaccine that's formed and used in the United States is killed or inactivated microbes. A killed microbe cannot replicate inside the human body. That way, they present no risk at all for infection or causing any symptoms as a result of the pathogen. However, they're not as effective at stimulating long-lasting immunity. That's why many of these killed microbe vaccines require multiple doses to achieve and maintain protective antibody levels. This is in the form of booster shots. The Salk or inactivated polio vaccine and hepatitis A vaccine are examples of this type of vaccine. The third type of vaccine that's used in the United States today is toxoids or subunit vaccines. Diphtheria, tetanus, and botulism result from the production of potent microbial exotoxins. These are inactivated by heat or formaldehyde, resulting in the loss of their toxicity, but they can still activate the immune system. The inactivated toxins are referred to as toxoids. The diphtheria, tetanus, and pertussis vaccine contains these toxoids from these various microbes. Hepatitis B, HPV vaccines are two vaccines that use toxoids that have been genetically engineered through recombinant DNA technology to be even more effective. There are other experimental vaccines, but they are not in use in the United States today. After talking about active immunization, let's now talk about passive immunity. Passive immunity is when the recipient, the patient, receives ready-made, preformed antibodies. These can be from other humans or animal sources. Passive immunity can include things like antitoxins or antivenin. Antitoxins are antibodies that are produced in a person or an animal after exposure to the toxin. Antivenin, similarly, is often from animals, not people, that have been exposed to venom of various snakes or spiders. In these cases, after brief exposure to the toxin or venom, blood is taken and processed. The antibodies are removed from the blood, and that is what is given as part of passive immunity. A big advantage to passive immunization is that the antibodies are delivered immediately upon injection and can be of life-saving value because they work so quickly. Immunity is received, but it is short-lived because it does not stimulate the production of memory cells. So there is no immunological memory. Immunotherapy can be hazardous because of the possible complications arising from the fact that the antibodies are often seen as foreign proteins by the recipient's immune system. This results in antibody production against the foreign protein in their serum, causing a condition known as serum sickness. This is almost similar to being allergic to the injected antibodies. The use of human serum taken from individuals after vaccination or during their convalescence period from a specific disease minimizes but does not eliminate completely the risk of serum sickness in the recipient. Next, I want to briefly cover a case that your book brings up and I think is an important lesson, in which an individual has sustained a puncture wound and is at risk of tetanus. Should that person receive active immunization by a booster shot with a tetanus toxoid, or should they receive passive immunization with tetanus antibodies? It depends on the person's immune history. If the person has, within the last 5 to 10 years, received tetanus toxoid vaccination, all that is necessary is another booster to effectively stimulate those memory cells already programmed in the body to produce tetanus antibodies immediately. However, if the individual has not received past immunizations against tetanus, the better thing to do is to provide that person with tetanus antibodies. That's because the amount of time that it would take for the person to generate tetanus antibodies on their own with the first encounter would be too long to save that person's life. Next, let's talk about vaccine safety. Vaccines have greatly reduced the burden of infectious diseases around the world, and as we mentioned, reduced the number of childhood deaths in the United States by 3 million every year. However, these diseases, because they are declining, have been almost completely eliminated from human memory. There are very few people alive today that remember how detrimental smallpox could be. And very few people in the United States knows how awful diphtheria or polio can be, leading to lifelong debilitations, living an entire life in an iron lung due to paralysis. Because these diseases and the memory of the deaths and symptoms of these diseases have been almost completely eliminated from society, people are now spending more of their time thinking about the vaccines themselves and how safe they are. There is no vaccine or any other medication that is 100% safe and without risk. In fact, if you think about it, even pure water comes with risk, as there are people who die every year in marathons from drinking too much water. So the question isn't to be asked, is it 100% safe? Because that just doesn't exist. The better question to be asked is, do the benefits of the vaccine outweigh the risks? In some cases this is true, and in other cases it's not. For example, oral polio vaccine is a live vaccine preparation that carries a vaccine-associated polio, so you can actually get polio from the vaccine in about 1 in 2 to 3 million doses. Now that's extremely small. However, in the United States, where we don't have polio, by giving this type of vaccine, you're actually incurring more of a risk from the vaccine than from not having the vaccine at all. In other areas of the world, getting polio is almost a guarantee, and in those places, there is a 1 in 200 risk of paralysis from the infection. In those cases, it makes far more sense to get the vaccine than to not. For this reason, when people in the United States acquire the polio vaccine, they don't use the live oral polio vaccine, and instead use the inactivated Salk virus vaccine. It does cost more and does not protect against the polio virus growing in the gastrointestinal tract, but it does prevent the paralysis that could cause as a result of the disease. It does not come with the risk of vaccine-associated polio, though. And because polio is so rare in the United States as to be non-existent, it is far better to get the inactivated Salk virus vaccine versus the Sabin live attenuated oral polio vaccine. In other areas of the world, that equation is the reverse, and the Sabin live attenuated oral vaccine is still given. Vaccines are constantly monitored and modified or withdrawn as certain circumstances dictate. So for instance, in 1979, when smallpox was declared completely eradicated from the world, smallpox vaccine was no longer given because, as we mentioned, every vaccine comes with a risk, however small. And when there's no smallpox available in the natural world, then the vaccine is no longer needed. This is, of course, unless you're in the military, where smallpox may still be a risk due to bioterrorism threats. Taking another example, in 1976, Fort Dix, New Jersey was threatened by the appearance of a new and deadly strain of swine flu virus. In some cases, the vaccine was associated with an increase in the risk of a neuromuscular disorder called Guillain-Barré syndrome. Guillain-Barré syndrome does sometimes form as a result of flu virus vaccines. However, when you take a look at the Guillain-Barré syndrome risk that comes with actually getting the flu virus, it is far more likely that a person would get Guillain-Barré syndrome after getting the flu than after getting the vaccine itself. But the CDC is always monitoring these statistics to make sure that the vaccine is safer than the virus or microbe itself. In another instance, in 1998, rotavirus was investigated. We'll talk more about this in the next section.